an extraction method to acquire adult insects for pheromone identification. And the second, which is funded by BBSRC priority call, is to go more after the pheromones. So they really are complementary to each other. So the general objectives for both projects are to optimize the method of collection of apple sawfly adults. Then there'll be entrainment of head-based volatiles coupled with the GCMS analysis to detect pheromone compounds associated with ASF. But we're also interested in collecting data on the reemergence of ASF, its distribution, phenology, the trapping of adults, and measuring the damage caused by larvae as the season goes on. And I'm just going to give a few updates of what we've been able to do so far. We've optimized and managed to actually nail down this flotation method for the collection of apple sawfly. It's adapted from method used in the Netherlands and has proven to work pretty well with our clay soil. The general procedure is we dig 30 centimeters deep into the soil below the tree canopy. We remove the surface vegetation, and then the soil is distributed in the trays. And then water is added and the samples left to soak. We are careful to remove large clumps of clay and break this down and then stir so as to release the organic matter. And any floating ASF cocoons are collected for our downstream work. And to date, I've been able to make collections at 4 sites in Kent, East Sussex, and Worcestershire. Over 160 apple trees have been sampled in and about. More than 480 trays, I'm sure there are more by today, of soil have been floated. And we've not been so lucky to get the thousands of ASF that we wanted, but we're making steady progress. We've got 70 so far. And these have been reared in individual pots in controlled temperature and light conditions at NIAB. So as you can see in the pictures below. And then we're also making steady progress towards beginning the monitoring phase of this project. We've already identified 13 or 15 orchards where trapping will be done. And I really want to thank our partners from industry and collaborators who have very happily volunteered their time and space for these traps to be set up. At NIAB, we've already procured the traps, and these are ready for dispatch by early next week for the monitoring to begin. We've developed a protocol for using these traps, and this has been shared with the partners. And really the plan is to hang these at about 1.5 to 2 meters height. It should be free hanging, nothing disturbing, and the traps need to be in the sun due to bycatch issues. So we've had a very progressive meeting with all the partners, and we shared all the tactics and techniques which we'll be using. And the traps will be set up before the pink bud green cluster emerges, and every week we'll get consistent data being reported by all the participating partners. And then we should be able to get a very strong data set that we can use for our work. I'm now going to move on to the second project, and then I'll take questions at the end. And this one is on forest bug. And again, what this project intends to do is to develop pheromones for the management of this pentatomic bug, which is an emerging pest of orchards in England. We've got a very nice collaboration with all these partners here, AgroVista, Avalon Fresh, Russell IPM, NRI, and of course ourselves. And these are the names of the people who are leading. Of course, the larger teams of other people support the work behind the scenes, and we appreciate them for what we're able to do together. The project aims are to identify the pheromone of forest bug and to produce novel species-specific traps for monitoring this emerging pest. And this work will pave the way for development of control strategies, which may include mass trapping or mating disruption. And all these are in response to the need to use less pesticides and still secure the future of apple and pear growing in the UK and beyond. We've made some progress since we started. Our partners and ourselves have conducted over 40 field trips, and over 500 forest bugs have been collected in the past year for rearing at NIAB. There is regular orchard monitoring that continues, and assessments of forest bug presence in Kent, Hampshire, Hertfordshire, and other regions as well. And AgroVista does manage an online platform so that we can be able to associate the weather data with what we're finding in the fields. So on the right is a chart just showing the general phenology that we've so far been able to get from when we started collecting last year. And you can see from as early as April, we begin to get the L2 and L3 life stages, all through to when adults begin to emerge. And this is one component which will be strengthening in this new year. We want to saturate this further because we do know that they do overwinter at the same time as L2s, but the emergence is more or less in cohorts. And so we really want to capture this in the field and how they emerge and how these different life stages emerge as the months go on. We've also had to overcome a couple of technical challenges. The first one was, how do we feed this forest bug in a way that we can rear them to adulthood? And this is how we started on the left with apple shoots and fisher eggs and protein sources and sugar. And it worked really well. And Michelle and Sterling really tried different optimizations. This was really cool. And then we had this meeting whereby it turned out that a much simpler and probably more effective way would be to use apple slices and really no need for protein source. And so this is what we're going to adapt for this coming year. It's also good to note that there's no need for temperature control or artificial lighting. And so we'll be adapting some of these changes into the rearing this year. We're hoping that if we missed something in the previous year in terms of the fitness of a forest bug, this year we should be able to produce a much more fit cohort of forest bug trees and experimentation. We also did sexing and produced a protocol to enable the identifications of males and females while they're still alive. And this has really been helpful in enabling us to do the entrainment. So we now know whether it's a male bug that we have in there or a female one. And we've done quite a bit of entrainments of both male and female forest bug in different combinations, single ones, males and females together, or females or males. And we've done over 93 entrainments, which then we've passed on to NRI for the GCMS and GCEAG. So the next couple of slides are really slides from our collaborators at our partners at NRI. And they've done over 43 analysis in different combinations. So for example, as shown here, we've had apple shoot female. This is a single female on an apple shoot. Over 12 of these have been analyzed, males as well. We've done blanks so that we can be able to remove that which is associated with the plant. And we've tried different systems to try and get the pheromone that is responsible for attraction. So far, we've not been successful in identifying from the VOCs that we've collected a pheromone. We've done both male and female, as you can see from this trace. We do detect stuff, but we just don't get a pheromone out of it. And there's no real difference between male and female. And our colleagues went ahead to validate their techniques using Nazara. This is another pentolid bug, just to be sure that the methodology they were using was working, and they were happy to confirm that what they're doing is working pretty well. And so we then think it's a matter of time before we're able to get progress with the forest bug. But in addition to that, they've also done analysis of whole body extracts using hexane. And really the idea behind this is to find out what are the cuticular hydrocarbons and if this may be involved in close range mating behavior as part of our experimentation. And we started this rather late in the season, so only 3 insects were subjected to this. But in this new year, we'll be doing a lot more with NRI on this. But in addition to that, the team at NRI has also been participating in the formulation of potential repellents and the synthesis of minor components. And we are also actively looking back in literature to find clues that we may get in there. What could be attractive, for example, to nymphs, at what concentrations, and how can we exploit this in the work that we do moving forward. But further to this, we've also been engaging other research groups and other people who work on pentatomid bugs and on forest bug. And so in January of this year, led by Michelle, we convened a meeting of a UK-EU forest bug working group. And we had representations from Belgium, Germany, and Switzerland, and we took time to share ideas and tactics and methodologies, and just, it was a very nice learning experience. Some of the things which did come up was they are observing up to 100% damage in organic pear orchards, which further underscores the seriousness of this pest. And that it's also hard to differentiate between the damage caused by brown marmorated stink bug, which is another petatomic bug, which I'll speak about in my second presentation later today, and forest bug damage. We also discussed about monitoring and where we could achieve the highest numbers. And clearly, they say that the trees with the rough bark seem to be more productive. We noticed the same during our own fieldwork. Usually, the older trees with the rough bark and then with new shoots at the top tended to be more productive in us getting forest bug, which you really need for experimentation. It also turned out that working the tree with a rubber mallet and having a sheet at the bottom is quite an effective way of disengaging them from the tree. So we've adopted that into our methodology. In terms of trapping, it turned out that it would be more beneficial to have a clear plastic trap on a wooden pole away from the foliage, and this was really good as well. We also discussed the prospect of acoustic communication, and time allowing, I could show a video on how these insects vibrate and whether this could be potentially, a short distance signal between the insects among themselves. We still believe that the pheromone is there and that is a long distance signal that they use to aggregate, because we've seen pictures sent to us of aggregations of these insects. But what that has done is caused us to observe closer what we saw, and we know that the L2 nymphs do aggregate in cages, and so we really want to investigate whether there are aggregation pheromones for the nymph forms. So our key priority areas, sorry, that has jumped, for this coming year are to strengthen in-field phenology data on when the different life stages occur from L2 all through to adult forms. We'll also be going deeper and more strategically into entrainments and the GCMS and GCEAG analysis. And this will be done for each life stage of the nymphs. And even when the insects get to adult form, we're going to do the entrainments at different ages. Hopefully with a new diet, we should be able to keep the adults going for up to 30 days and then see probably it's at the early stage or the mid stage of their adulthood or whatever stage, we are going to find out for sure which at which stage we get pheromones. But in addition to that, we are very keen to provide solutions to the growers. So we'll be identifying and sourcing traps for field testing, and this will include the Starling rescue trap, flea and sticky light traps, and a variety of other field tests during the year in collaboration with our partners. We'll also be doing field experiments using virgin male and female forest bug on a pole away from the canopy. We did do this experiment last year, but we had the trap within the canopy. This time we're going to have it more in the open to see whether it's a male or the female which has got the attractive pheromone. And then we're also going to explore possibly the use of repellents as an alternative to having an attractant. So thank you, and I'll now take your questions.
Thank you very much, Francis. Very helpful. And I've got one or two for you here. Regarding the repellents, how are NRI approaching that, and what sort of repellents are they choosing? And is their choice based upon previous research they've done with other insects?
Yes, at the moment, that's the approach which we are taking. We are looking a lot at the literature, what has been found in there, and is that something which you can possibly exploit? Of course, it's a bit of a diversion because the original idea is really to get a pheromone, and that's really where we're trying to still have our main efforts in.
Yeah, okay. And another question I was thinking is, do we know, or have we got much evidence to show that the numbers or populations of forest bug differ in different regions of the UK? I just wondered, because you've been collecting samples from different regions, do we have any gut feeling about that? Maybe that's a question for the agronomists, I don't know, but any views from you?
Yeah, probably, yeah, maybe that's one for the agronomists, to be honest. But even within the same region, we notice that orchards have got different. Some orchards are really productive, some are not, and in the same region. So, but yeah, that's something which I think we can
when you say productive for forest bud rather than
for forest bud, yes, which
yeah.
maybe not necessarily a good thing, but
okay. I don't think we've got any more questions. I should have just said, and I apologize, forgive me for not explaining that Francis is a member of Michelle Fountain's Pest and Pathogen Ecology team at NIAB at East Malling, and so he's leading a lot of this work at the moment. So Francis, thank you very much for sharing that with us this afternoon.
Thank you.
So we're now going to move into some presentations from our PhD students. Many of you will know that NIAB at East Malling has a number of PhD students. We have 3 that Michelle Fountain is mentoring at the moment, also in collaboration with University of Reading. And the first of these is Charlotte Howard. And I've asked each of the students whether they're just giving us 10 minutes updates on some of the research that they're doing within their PhD studies, which will be of use and interest to the tree fruit industry. The first of the 3 is Laura Reeves, who's based at the University of Reading, but as I said, collaborating with us at NIAB at East Malling and doing some work here as well. Laura is not able to join us this afternoon, but she has kindly recorded a presentation for us. So my colleague Arunima hopefully has got the presentation ready for us, and she's going to share that with us now, and fingers crossed all will work. It looks like we've got some activity. There we go.
I'm Laura Reeves from the University of Reading, and my talk today is on pear sucker and their natural enemies. For my project, I'm looking at the interactions between pear trees, pear sucker Cacosilla piri, and its natural enemies. It's a good model system, as C. piri is the dominant pest of pear in the UK, with a large amount of information on its life cycle and natural enemies. It goes through 5 nymph stages and has an adult winter form, which is larger and darker with smoky wings, and a summer form, which is smaller and lighter with clear wings. Its natural enemies include anthracorids, earwigs, ladybirds, and spiders. However, there are concerns over climate change altering trophic interactions due to change in feeding habits, activity levels, and phenological mismatches. During this presentation, I'll mainly be talking about the interactions between the anthracorid Anthracoris nemoralis and pear sucker. A. nemoralis is the main natural enemy of pear sucker in the UK and Europe. A single anthracorid is estimated to consume 5,000 pear sucker eggs during its lifetime. Natural migrations of anthracorids can reduce summer pear sucker populations, peaking during July and August. Anthracorids can also be released artificially into orchards as a biocontrol agent to reduce pear sucker populations more rapidly. Therefore, it's important to monitor how climate change can impact such an important natural enemy. However, there's concerns about the influence of rising temperature on insects. Insects are polykelothermic. They have a body temperature that fluctuates with their environment. There's evidence that pear sucker has temperature-dependent development, where faster development rates at warmer temperatures could lead to shorter generation times, increasing pest populations. There's also evidence that feeding rates could increase due to a higher metabolic rate under elevated temperatures. This is also influenced by body size. So smaller pest species could have a larger increase in metabolic rate than larger natural enemy species. However, more studies are required on A. nemoralis, monitoring their behavioural and functional responses under elevated temperatures. So what is a functional response? A functional response is the change in a predator's consumption rate, so the number of prey items eaten, depending on prey density, over a certain time period. It consists of the attack rate, so the rate of discovery of a prey item, and the handling time, the time taken to consume a prey item, including the time taken to capture, kill, eat, and digest it. If you look at figure 1, there are 3 main types of functional response. Type 1, where there's a linear increase in consumption rate depending on prey density, and the time needed to consume prey is negligible. Type 2, which includes handling time, and at low prey densities consumption rate increases, but remains constant at high prey densities. And type 3, where at low prey densities consumption rate increases slowly due to learning time, as the predator needs time to recognise a prey item. Our 4 hypotheses were that adult anthrochorids demonstrate a type 2 functional response, that handling times are shortened and attack rates increase at higher temperatures, behaviours including movement, antenating, so the movement of anthrochorids' antennae, and cleaning in anthrochorids increase under elevated temperatures, and that the sex of anthrochorids impacts the functional response, with shorter handling times and higher attack rates for females compared to males. So females consume more prey. The methods used for our functional response experiments were we starved adult Ani moralis for 24 hours in controlled temperature cabinets, which were set to either current mean summer temperatures or predicted mean summer temperatures for 2080 based on low or high emission scenarios. The anthrochorid was then added to a petri dish containing 1% agar, a leaf disc, and pear sucker nymphs at one of the 5 different densities. After another 24 hours, the number of dead and live pear sucker nymphs were counted to see how many the anthrochorid had consumed. There were 10 replicates for each treatment and a control to account for natural mortality where an anthrochorid was not present. We then fitted functional response curves using the Fryer package in R. For our behavioural response experiments, we also starved anthrochorids for 24 hours in controlled temperature cabinets. The anthrochorid was then added to a petri dish containing a piece of leaf with one of the 3 treatments on, with pear sucker eggs, pear sucker nymphs, or no food present. The anthrochorid was given 10 minutes to acclimatise, and then the movement and behaviour of it was tracked for 20 minutes using ether vision software. The room was kept at one of the 3 temperature treatments with 10 replicates for each treatment. And figure 2 is an example of the heat map of movement of an anthrochorid made using the ether vision software. The results of our functional response experiments were that a type 2 functional response was demonstrated for male and female anthrochorids at all temperature treatments, which can be seen in figure 3 based on the shape of the curves. Males demonstrated significantly longer handling times and lower attack rates than females, and on average 12 pear sucker nymphs were eaten by females at the highest prey density compared to only 8 nymphs eaten by males. Males also had a significantly longer handling time at 18 degrees compared to 23 degrees, and all other temperature differences were non-significant. Contrary to our second hypothesis, results from behavioural assays found that anthrochorids spent less time moving at 23 degrees, the highest temperature treatment, for egg and no food treatments. Anthrochorids also spent significantly less time antenating and cleaning at 23 degrees compared to 18 degrees in either egg or no food treatments, which can be seen in figure 4. However, no significant differences in time spent feeding or differences between males and females were found, although we only looked at 20-minute time periods to analyse anthrochorid behaviour. To conclude, the sex of anthrochorid impacts the handling time and attack rate of the functional response. Male anthrochorids had much longer handling times and lower attack rates than females, leading them to consume less pear sucker nymphs. Although male anthrochorids had significantly longer handling times at 18 degrees compared to 23 degrees, there was no temperature-dependent differences for females, which could be due to body size, as male anthrochorids are much smaller than female anthrochorids, so could be more sensitive to temperature. Ani moralis is likely to be an effective predator of sea prairie nymphs under future temperatures, as there was no significant difference in feeding behaviour in the behavioural assays. However, there was a uncertain about the reduction in activity and cleaning behaviour at higher temperatures, as grooming is an important sanitary behaviour which could potentially elevate the disease risk under higher temperatures. There is a gap in the scientific literature about interactions between natural enemies of pear sucker. Earwigs and anthracourids are both important predators of pear sucker. However, monitoring data indicates that when earwig abundance is high in the summer in pear orchards, anthracorid abundance is low. I plan to do a microcosm study this summer to see if intraguild predation is occurring, so when earwigs consume anthracourids, or interference competition is occurring, so when earwigs alter the behaviour of anthracourids. Thank you for listening. If you have any questions, please email me.
So thank you to Laura for that presentation. Obviously, she's not with us this afternoon, but do please contact her. Or if you want to pass anything through me or do it through the chat facility, feel free to do that as well. But hopefully you'll find that a useful update. And it's nice that Laura's still got some more work to do in this coming year. And hopefully we can share that with you at future events like today. So, Laura is the first of our 3 PhD students. Our second is Charlotte Howard. Charlotte is also based at the University of Reading, but working with Michelle Fountain closely in the work she's doing on wildflowers. And many of you who attended the Bespoke event that we ran at East Malling back at the end of October will remember that the whole purpose of that day was to have a look at the impact of wildflowers on bees and pollinating insects. And so on this topic, Charlotte's going to be chatting to us today about what she's been doing on the impact of wildflowers on orchard pest control services. So Charlotte, perhaps you'd like to share your presentation with us. It's really helpful. So our third student of the 3 is Konstantinos Tsiolis, who's also working with Michelle Fountain and the University of Reading. And he's going to be telling us a little bit about nesting preferences of solitary ground nesting bees. Good afternoon, Konstantinos.
Good afternoon.
Hi, and yeah, if you'd like to share your presentation with us.
Yep.
And you can tell us a little bit about what you've been finding.
Absolutely.
That's perfect. We can see.
Yep, perfect.
Thank you.
Thank you very much. Great. So hi, everybody. It's a great pleasure to be here. So I just finished my PhD project and I'm currently waiting for my viva. The title of the presentation today is Nesting Preferences of Solitary Ground Nesting Bees in UK apple orchards. And this project was in collaboration with University of Reading, NIAB and Worldwide Fruit Limited. So, as we all know, 1/3 of the food we consume depends heavily on animal pollination. Its value has been estimated between 180 and 442 billion pounds worldwide. Studies in literature are increasingly highlighting the importance of wild bees to pollination of crops. A recent study has shown that the value of solitary bees to UK apple pollination is at 51 million pounds per year. And just for comparison, the value of honeybees has been estimated at 21 million pounds per year. So the survival of bees depends heavily on both food and nesting resources. For food resources, we have a fairly good idea of what we need to do to provide them, such as providing wildflower meadows in farms. However, when it comes to nesting resources, it's a very under-researched and poorly understood area. This is extremely important if we consider that 70% of bee species are actually ground nesters. So today I will talk about two of my chapters, so two of the studies of my PhD. The first study I will talk about has been recently published, and you can go and read it if you are interested and find out more about it. But the first reasons question was, what are the preferred soil properties of solitary ground nesting bees? Also, we were interested to find out whether bare ground plots can actually provide beneficial nesting resources for a very important ground nesting bee species, such as the apple pollinators, Andrina hamoroa, Andrina dorsata, and Andrina nitida. So 8 south-facing bare ground plots were created in 2018 at NIAB East Malling. The vegetation, the dimensions of each plot was 10 meters by 2, with 1 meter 10 degrees slope. And you can see an example of a plot at the bottom left of your screen. The vegetation cover was controlled via herbicide treatments, and then surveys took place in 2018, 2019, and 2020 to record attracted species and nest density of each plot. Then 9 soil variables were measured, which is soil compaction, hydraulic conductivity, organic matter, root biomass, stoniness, soil water content, soil texture, soil temperature, and vegetation cover. I was extremely excited to find that bees actually nested in the plots, and as you can see, the nest density varied between plots. But just to give you an idea, in one plot of 10 meters by 2, I have counted 550 nests. That was the highest number of nests I counted in a such plot. So in terms of which variables are actually determining nest site selection, this study has shown that hydraulic conductivity and vegetation cover were significant negative predictors of nest density, where soil stoniness and soil temperature were significant positive predictors of nest density. In terms of which species have been attracted to nest in such plots, 870 bee specimens were sampled and identified, which belong to 26 bee species. 11 of these species were kleptoparasitic, and the most abundant bee species was Lasioglossus malacurum. I have also recorded 13 ground nesting wasp species. However, it was very interesting to find that no Andrina species of interest were recorded utilizing such plots. So then the question was raised, if they do not seem to prefer nesting on bare ground, fully exposed bare ground, where do they nest? This led to the design of another study, which aimed to determine whether these 3 important apple pollinators are actually nesting in apple orchards. If they nest in apple orchards, what is their preferred orchard habitat? Alleyways, under trees, edges, and then also, what is their preferred local habitat in terms of vegetation cover? Do they actually prefer nesting in vegetation if they do not prefer bare ground? So during my PhD, 17 apple orchards were intensively surveyed in Kent, where 2,000 square meters transect took place on south, east, and west facing edges of apple orchards. Also, 2,000 square meters transect took place in alleyways and under trees of each orchard. So once a nest was found, it was marked and labeled. Then the nest entrance of the nest was measured because these ground nesting bees come in different sizes. So by measuring the nest entrance, it can give us an idea of which species might be nesting there, or which genus might be nesting there. Then habitat assessment took place 20 centimeters around the nest entrance. And then the nest entrance was covered with a conical trap, which you can see on the right side of the screen. And here I need to give credit to Michel Fontaine and Celine for coming up with this idea, which is basically a strainer that they're using in bars for cocktails. And we decided to put it upside down, make a hole on the top of the cone, and then cover it with a plastic tube. And it was a very effective trap, as you can see on the photo. So recorded traps were visited every 20 to 30 minutes, and recorded specimens and trapped bees were recorded, or several photos were taken for later identification if it was not possible to identify them in the field. Some other species, as you can see, took advantage of these traps. However, it was in very low numbers and it was not considered significant. I think only 3 cases I have recorded this. So yeah, nothing to worry about here. In terms of results, orchard and local habitats were significant predictors of nest density. As you can see on the graph, bees mostly prefer to nest under trees and edges. And in terms of species, several species which you can see on the graph have been recorded nesting in apple orchards, including our species of interest, Andrina hamoroa and Andrina nitida, which mostly nest in under trees and edges of apple orchards. In terms now of local habitat, the long vegetation and short vegetation was significantly lower in nesting habitat than control habitats, where moss and bare ground were significantly higher in nesting than control habitats. That's when things got extremely interesting, even more interesting. So one day I had a sandwich by a hedge of an apple orchard, just having lunch, and all of a sudden I could see bees diving into long vegetation. So the vegetation on the left, on the photo on the left side of the screen is approximately 80 centimeters tall. So I could see bees diving into vegetation, and this got me very excited. I got out of the car, and long story short, I was able to find several bees, several nests under such long vegetation. So each red flag here represents a nest or more. So then I decided to revisit several others apple orchards to see whether I can actually find this nesting behavior elsewhere. I found it in another orchard and then another. So overall, this was recorded in 3 different apple orchards. And here I included some videos just to show you how visible these nests can be. On the first video on the left, you can see a semi-vegetated habitat, mostly thatch and bare ground, which is very easy to see the nest entrance with some experience. However, on the right is an example of a nest under long vegetation, and I was only able to actually suspect that might be a nest there by seeing the bees, bee diving into the vegetation and then moving the vegetation around to find the nest. So as I said, three such nesting aggregations were recorded. So I decided to take advantage of this finding and investigate this nesting behavior even further. I need to mention that the great majority of nesting species were the important species of interest for apple pollination, Andrina hamoroa and Andrina nitida. So I decided to have two control habitats per apple orchards where no bee nests were recorded, and then my nesting aggregation. 10 sampling areas were determined from each control habitat and nesting aggregation. And then the nine soil variables I previously mentioned for the first study were also recorded here, as well as a hedge type, as these aggregations were along hedgerows. So in terms of nest size selection of this aggregation, so why these species seem to prefer nesting in such places? I found that soil temperature and soil stoniness were significant positive predictors of nest density, where soil moisture was a significant negative predictor of nest density. Now, my conclusions. Nesting resources are very critical for the sustainability of ground nesting bee populations. My first study has highlighted that appropriate bare ground habitats can be a very useful nesting resource for several bee species. My second study has shown that bees mostly prefer to nest under trees and edges of apple orchards. Also, it has shown that bees have mostly preferred nest in bare ground, moss, and short grass. We suggest that farmers will need to conserve and increase bee nesting resources in apple orchards. And in the near future, I aim to produce a list of recommendations of what farmers need to do to be able to achieve that. A recent study, which University of Reading was involved, has highlighted that pollinator diversity is very important for interannual stability in pollinator communities. We suggest that by creating both bare ground habitats and semi-vegetated habitats, we can provide such bee diversity in apple orchards and ensure sustainability of pollination in the future, especially when we consider climate change and many other factors. However, my PhD has only touched the surface of the nesting preferences of ground nesting bees, and further research is needed to be able to enhance our knowledge in this very important area for pollination of our crops. So thank you very much for your time, and I'm more than happy to answer any questions you might have.
Thank you, Konstantinos. There are one or two questions filtering in the chat, but given that we have gone quite heavily over time, I think we will move on. But if you would be kind enough to answer the questions in the chat facility, that would be very helpful. We must move on to our final speaker, and we're working Francis Wamonje very hard this afternoon because he's kindly agreed to come back and tell us a little bit more about brown marmorated stink bug and what we're learning about that new invasive pest. So Francis, if you're out there, can you share with us again, please? Yeah. Thank you, Konstantinos. Okay.
So just in a second.
Do you want to share your presentation?
There we go.
We're slightly limited on time, Francis, so if you can read brief, that would be great. Thank you.
Great. So this is going to be a short presentation on work we did for 2022 for brown marmorated stink bug. So just a brief background. BMSB is an invasive pest established in many European countries. There have been reports of interceptions in the UK since 2010, and since 2018, adults have been observed in the UK. Now, concerningly, since 2021, both male and female individuals have been trapped, and therefore this project really conducts surveillance in the UK to find out if detected adults represent hitchhikers, or if the pest may have already or is at the initial stages of becoming established within the UK. And surveillance efforts have mostly been in regions considered climatically suitable for the establishment. So mostly Greater London, East Anglia, and the Bristol Channel coastal areas. And how we do this is using a network of pheromone traps hosted by multiple volunteers. And we do beat sampling, especially where sightings have been confirmed. And what I'm going to really concentrate on are the findings for 2022 in this presentation. So this is just a summary. We did surveillance between May and November 2022. That was last year. And there was additional surveillance, which was independently led by Dr. Glenn Powell at RHS Wesley. Some of you may know him. And we had double the number of sites deployed in 2021. So we had 3 sites in Bristol, 4 in Norwich, 6 in London, and 17 across various sites in Southeast England. And Glenn Powell set up 6 sites at RHS Wesley. We also had a very nice collaboration with the Natural History Museum, who set up quite a number of traps for us within London. But further to that, we also have got a public reporting channel. So there's a dedicated email, which we monitor very, very regularly. So whenever people spot a bug which they think is brown marmorated stink bug, they do send a picture. And from there, we are able to confirm whether it's a sighting or not. And in fact, this is how the first sighting of 2022 was achieved by a member of the public. I think the first one was sighted around the Parliament area in London. We also conducted beat sampling at different sites at RHS Wesley, where consistently been getting 1 or 2 catches in the past several years. So this table is essentially a map showing the sightings since 2018. And you can see from 2018, a single sighting, 19, and the numbers begin to increase in 2020. And 2021 is really when we begin getting males and females noted. Now in 2022, you can see that in our pheromone traps, entries number 22 and 25, we're getting both male and female caught within the traps. And so in a way, we know this is a good confirmation that our strategy is working, but also concerning because when these begin to turn up in pairs, then it's only a matter of time before we confirm an establishment within the UK. And the last sighting, which was reported by Andrew Sainsbury, who works at RHS, was associated with a returning camperone from the continental Europe. So again, this goes further to just confirm that our strategy of looking at camperone traps and having camperone areas and having traps in those regions is a good surveillance strategy. And on this final slide, I'm just sharing pictures which were taken by Glenn Powell. And this was a female on panel A, which was caught in one of the pheromone traps that you can see in the background. And it laid eggs, but this one viable, so it was probably unmated. But pretty soon, a day after or so, a male was caught in the same trap. And these were both recovered. There was no further mating. But this is the kind of thing which tells us that 1, our surveillance strategy is working, and 2, that there is need for us to be concerned about the establishment of this pest which affects many tree fruit. Right, I'll take your questions.
Thank you so much, Francis.